Hello everyone, my name is Spencer.
I'm Kevin.
I'm Zia.
And we're going to be investigating the inhibitory effect of bioflavonoids on Escherichia coli as a response system.
So just a little background on our project. We're going to be looking at the SOS response system in E. coli. And what this system does is it repairs and detects DNA damage in the bacteria while it's under stress. And this system is regulated by two proteins, REC-A and LEX-A. And this is the pathway that the SOS system falls under. As you can see, when DNA damage is detected inside the bacteria, then REC-A is expressed, which in turn cleaves the LEX-A repressor. LEX-A is a protein that controls the SOS response system. It represses the SOS response system. So, in a nutshell, when REC-A is expressed, then the whole SOS system is functioning. By its nature, this SOS response system is error-prone. So when the DNA is being repaired, mutations can occur which can result in things such as antibiotic resistance.
Bioflavonoids are plant-based organic compounds, also known as vitamin Bs. They have been known to exhibit anti-inflammatory, antimicrobial, and anti-ulcer effects. They can be found in natural sources such as celery and apples. For research, we're focusing on two main substances of bioflavonoids, which are flavonoids and flavonols. As you can see, the main difference between them is that in flavonols, there's a hydroxyl group on the surface of the central ring.
In this experiment, we are going to be using E. coli, which is a gram-negative, rod-shaped, and BSL-1 bacteria. E. coli is usually found in the lower intestines of cold-blooded animals. Although it's usually harmless, some strains of E. coli may cause gastrointestinal infections. Therefore, in our experiment, we will be using E. coli K12, which is a safe strain and won't cause these diseases. E. coli K12 is also cheap and readily available.
We conducted a literature review and found that in a 2011 study in China, Kailin, which is a flavonoid, was able to inhibit the SOS response system in another bacteria known as Staphylococcus aureus. In the same study, it was also found that the inhibition of SOS response system showed a lower mutation rate. In another 2004 study in Japan, it was found that the anti-inflammatory effects of bioflavonoids were affected by the provision of hydroxyl group.
So Mr. Wolf always likes to say, how do we save the world with our experiments? Well, it's truth that antibiotic-resistant organisms are on the rise. Since the 1990s, the number of resistant organisms have increased, while the number of drugs that FDA approves every year are decreasing. So if you put those two trends together, you run into a problem in the future where our antibiotics are not able to treat our antibiotic-resistant organisms. So by conducting this experiment, by looking into the potential inhibition of SOS response system by bioflavonoids, we could enhance the effectiveness of our antibiotics that we have today. So an objective from the Academy of Science side, we'll be investigating if pure flavonoids, which we mentioned was a large subclass of bioflavonoids, can inhibit SOS response system in E. coli K12.
For the ACI side, we'll be investigating if flavonols, which is another subclass of bioflavonoids, can show the same inhibitory effects.
So in this experiment, our hypothesis for the AOS side is that more DNA damage will be measured in E. coli while it is exposed to flavonoids under UVC irradiation. This is because if these flavonoids truly do inhibit the SOS response system, then no DNA repair will occur, which results in a higher measured DNA damage. This DNA damage will be measured using single-cell gel electrophoresis, also known as common assay.
Similarly, for the HCI side, we hypothesize that more DNA damage will be present if E. coli is exposed to flavonols under UVC.
In the AOS side, our independent variable will be the type and concentration of flavonoids. Flavonoids are a subclass of bioflavonoids, and we're going to choose 3 different types of flavonoids in 3 different concentrations. These concentrations will be decided by our toxicity assay. For the AOS side, we will be measuring the extent of the double-stranded DNA damage using common assay. Likewise, for the HCI side, we will be varying the type of flavanol. We will choose 3 different types of flavanols and 3 different concentrations previously mentioned. Also, they will be measuring the extent of the double-stranded DNA breaks. Controls in our experiment include the type and strain of bacteria. Both groups will be using the strain E. coli K12, as well as the wavelength of VEC light, which is 254 nanometers. Both teams will dissolve their bioflavonoids in the same solvent, which is dimethyl sulfoxide, also known as DMSO. We will keep our bacteria at the same temperature, and we will keep the duration of the exposure constant between our 2 groups.
Since we hypothesized that after adding bioflavonoids, E. coli will have more DNA damage, our positive control group will have E. coli without bioflavonoid exposed to UVC. This will create more DNA damage. The common assay will test the amount of DNA damage. The length of the tail shown in this diagram will will show the extent of the DNA damage. This is the expected result for a positive control group, where there is a relatively long tail which shows more DNA damage. On the other hand, for a negative control group, E. coli will be cultured with bioflavonoid without UVC. Therefore, there won't be any DNA damage, as shown in this diagram, where there is only the head and no tail.
So we chose these 6 chemicals for very definite reasons. A few of the reasons. One of the reasons is that the price. These bioflavonoids have been known to be expensive because of their extraction source. So we chose these chemicals that were cheaper than most bioflavonoids. Second, if you can see, bicolony and chrysamin, which in our side is a flavone, share a similar chemical structure. In fact, the only thing that's different between those 2 chemical structures are the addition of hydroxyl group on bicolony. Also, diosmin has a maltose group attached on its chemical structure. And when we do these comparisons, we could possibly look at the effect of hydroxyl groups when we compare our results from bicolony to chrysamin to diosmin. On the Hua-Chang side, we can look at the morin and quercetin because it also shares a similar chemical structure. In fact, the only thing different between them are the position of the hydroxyl group on the right benzene ring. Also, rutin has a maltose group, much like the diosmin, on the right ring. So we could, when we compare our results, we could further look into whether or not the maltose group has an effect in our inhibition.
For our method, firstly we'll be making solutions of bioflavonoids. The bioflavonoids we have chosen are non-polar, and so we'll be using DMSO as a solvent to dissolve them. After which we'll be throwing E. coli kit and conducting our toxicity assay to find out the minimum inhibitory concentration of bioflavonoids and minimum inhibitory duration of UVC exposure. After which we'll expose the E. coli to UVC radiation before growing them to bioflavonoids for a set period of time. Finally, we'll conduct our COMET assay to measure the extent of DNA damage to see if the bioflavonoids have successfully inhibited the SOS response system. We choose to use COMET assay because both AOS and BCI are able to conduct the COMET assay, and also because it's more cost effective as we have a high number of trials as well.
So there are some obvious limitations with our project. We're only looking at one strain of bacteria, E. coli K12. So even if our experiment shows that the SOS response system was inhibited by these bioflavonoids, we don't know about the other bacteria that these will. Also, we're only looking at 2 subclasses of bioflavonoids. There are 8 classes total. So even if we find that these bioflavonoids that we have chosen are shown to inhibit SOS response, we don't know about the other classes.
Another limitation is that we are going to use COMET assay to measure the amount of DNA damage, which is not a direct measure of protein expression. Also, we are going to use UVC light instead of antibiotics to create DNA damage.
So when we bring our 2 results together, we can tie them together and then try to answer some of the questions we posed. Our main objective is whether or not flavonols and flavones can inhibit E. coli's response system. While those are only 2 subclasses of our 8 subclasses of bioflavonoids, those 2 are the largest of the bioflavonoids. So from that we can move on to a future application where all the bioflavonoid classes could be tested. Also, we mentioned the 6 chemical structures that we are choosing. We chose those chemical structures because they differ only slightly by their position of the hydroxyl group and the maltose group. So those things could have an effect in our inhibition. And I guess the last thing that we're going to say is that we are doing this to combat antibiotic resistance. That's a real problem in the world where many people are dying. In fact, 70,000 people are dying every year because we're not able to treat such resistant organisms. So can bioflavonoid-based drugs be an ultimate solution to combat these antibiotic resistant problems? So references. Thank you.
I have some questions. Just a couple of comments on the presentation. First of all, the idea of doing a literature survey, where'd that come from?
We realized that our study has a lot of assumptions that people could make, and we want to make sure that there is scientific research on the assumptions that we could be making.
I love that. I thought that was very cool, that you put, I mean, people often talk in their presentations, in fact, I could be wrong on this one, but have we ever had a group that said literature survey at the top of their slides before? I just thought that was kind of cool, you know, because it really showed that here's what's been done, and it's been done in the literature. So I thought that was a real strong point. Sequence on this. So you'll hear me say a lot this year, and you'll hear your mentors say, tell the story. Tell the story, okay? This is a good story. And be careful the way you sequence it. So for example, Kevin, you mentioned comet assay 3 times before we described what it was in their presentation. And you were you said, we're going to use the comet assay, we're going to use the comet assay, we're going to use the comet. That should have been, you should have said, we're going to use the comet assay and here's where it is. Because you don't ever want to assume that the audience knows what you're talking about up to a point. Okay, so you don't want to say DNA and here's the double helix. And this is what it looks like, and this is A, and this is T, and a positive. You don't, so up to a point. The other thing in terms of sequence is you got very passionate when you started talking about your rationale.
Yeah.
That should have been first.
I know.
I thought, that was awesome. It was here's why we're doing this. Why not start out with that? And one last thing about the sequence, you're doing much more here than, I think this is a very good experiment. You're doing a whole lot more than testing antibiotic resistance. And you said that in your very second or third slide when you had that whole sequence of, put that second slide up there. The Rec-A thing. Don't trivialize this diagram. This is hot, okay? You're looking to see a spot, and ultimately you may take this to the point where you may find out where in this metabolic pathway this thing is stopped. So I was waiting for this slide to come up again at the end as kind of so here's what we're thinking of doing sort of thing. And those are just storytelling issues. The science looks good. But you want to tell your story, in a really coherent, hook 'em way, made to stick. Now read that book again. Go ahead, I'm sorry, I'm done. If anybody else has any questions.
That was Grub.
Why are you using UV instead of antibiotic?
Well, we're not actually allowed to create antibiotic-resistant bacteria in our lab because that would be a safety issue. So we use UVC light to simulate those antibiotics and create damage.
And there's best research that's been done that as a response system, it's not only induced by antibiotics, it's induced by a lot of stresses that could cause DNA damage, and UVC is one of those stresses that can induce as a response system.
And what kind of UV source are you going to use, and how long are you going to install it to it? Are you going to, because if you install it too long, it'll die.
Exactly. So that's why we included that into our toxicity assay to make sure when we choose our duration of UVC light, we want to make sure that our E. coli is stressed, but it's not all dead.
Also, we're going to be putting the bacteria on Petri dishes inside the Gallic cabinet, which is our UVC source.
Say what?
The Gallic cabinets will be our UVC source.
Anything else? Yeah, Dr.
Dr. T.
Maybe you can tell me if I got this right.
Yep.
Your big idea was that you could maybe use bioflavonoids in conjunction with an antibiotic to both kill the bacteria but also prevent them becoming resistant?
That's our future application because bioflavonoids, if our experiment is proven, if more work is done based on our experiment, we could possibly see antibiotic resistance being inhibited by our bioflavonoids. So that would be a realistic potential solution to combating antibiotic resistance. Okay. Yeah.
So you might make it a little more explicitly clear because I think that's a good point. I don't know if it would be obvious to people.
That's kind of exactly what I was getting at. There's a lot more to this than seeing if you can kill bacteria. This is a very good study, and I think you're missing some of the real good science. Don't dumb it down. Don't dumb it down.
This is a suggestion not only for you and the rest of the team. See, this is a concept which is taken from theater. When 4 people are there performing something, if one drops, others have to just immediately step in to not show him off the rest of us.
Yeah. Save his part.
So God, you all know the story. It's not that you know only your part. You all know the story. Step in and take the cue from there and keep walking because nobody notices that something's happening. And
DNA is a very complex structure.
Yeah.
How would you be able to recognize the effect of the UV ray, UV radiation?
Common assay. Explain the common
Well,
assay.
okay, so that's basically what our measurement assay is, which is a single cell gel electrophoresis. And what it does is that basically in layman's words, after we stress our E. coli, we'll lyse it with the lysine solution and then basically take out the DNA and see if the DNA is floating in the buffer that we
How
have.
are you going to see it?
It's you look at the results.
Explain how the common assay
So
works.
this is a typical common assay result that we will get in conclusion. And the head is basically our cell. And if you see, I don't know if you can see it, but there's a tail, which is why it's called a common assay. And basically more tail, the longer the tail, more DNA damage has been inflicted. And how we're going to quantify this using a software in our public domain system. And it's basically measuring the length of the fluorescence, because this is going to be fluorescent. And then measuring the length and quantifying it to actual numerical data. Any questions?
Now, for me, thank you.